Hopefully that looks okay.
Yep.
Okay. So again, thank you for allowing me to be speaking today. I am Nancy, as mentioned, and today I'll be giving you a quick overview of the Reactome Knowledge Base, which is where I work as the Reactome Outreach Coordinator. And after I finish talking, Helia will take over and talk a little bit about her project. So for those who are not too familiar with Reactome, what we are is an open source, open access, manually curated and peer-reviewed pathway database. So we provide essentially pathway data for human reactions, and some bioinformatic tools for representation, interpretation, and analysis of that pathway knowledge. And we provide this for free for any user. So no matter if you're working at a lab or working in industry, you can use this pathway knowledge however you wish. And the only thing we ask for is some credit. So if you publish, if you cite us, then that's sufficient for our licensing. So our team formed this project in 2002. So we're now in our 22nd year of developing this pathway knowledge base. And we're led by 4 different PIs. So one is Lincoln Stein, who is my boss at the Ontario Institute for Cancer Research, and then 3 other PIs, one at NYU, one at the European Bioinformatics Institute, and one at the Oregon Health and Science University. So pathway diagrams, such as the ones that are in Reactome, are generally represented in a variety of different notations. So in our case, we chose the process description style of the systems biology graphical notation. So what this means is our diagrams represent all of the biomolecular entities and the mechanisms involved as a molecule that's consumed or produced through a reaction, and that reaction is mediated by either stimulatory or inhibitory molecules. So this type of diagram, as opposed to other ones, are directed, sequential, and mechanistic. And you'll see if you go to the Reactome website that each of our reactions follow that format. So in general, the way that things enter our database is through a manual curation process. So you can see that over this 22 years, we've accumulated many different human pathways, and that breaks down into over 15,000 reactions. And to support those claims, we have to go through over 38,000 literature references. So you can imagine that that's quite a difficult manual process. And overall, the process is carried out by an in-house curator that we hire as a staff scientist. So the curation process involves the curator identifying a pathway of interest and conducting a literature search. They compile all of the molecular mechanisms, reaction details, and connections to create that pathway. It gets placed into the database, reviewed by a different senior curator, and then it actually gets peer-reviewed by an expert researcher outside of our organization, so that it's very similar to how a journal has peer review conducted before things get published. And overall, that happens 4 times a year. So if you are interested in getting different biological information into the database, it does happen really frequently. So you can always contact our team if you have one that you're interested in helping us with. From the user side, the information that is captured in the database can be accessed in a variety of different ways, such as these high-level illustrations, which, again, if you're a researcher or if you're an educator, you can feel free to use these diagrams as you wish in your presentations and publications. We also show subpathways that are involved in that overall biological process, and then the individual pathway diagrams are in that systems biology notation that I talked about. We also provide written summations. So if you go to a pathway of interest, you can see something that a curator has written and has been peer reviewed that describes each step of that process. And it also includes the literature references that they've used. We also have some analysis tools and data representations for users who want to understand their own data set in the context of Reactome pathways. So very briefly, we have a few different types of analysis tools. So we have ones for overrepresentation analysis, which might be similar to a gene ontology type analysis that you may run, except in this case, we use Reactome as the scaffold. We also have pathway expression analysis, which can be accessible through the web and also through an R package. And we have some more advanced tools like network analysis and pathway prediction through our Cytoscape app. So I won't talk too much about the analysis tools, but if you have questions, please feel free to reach out. We're always happy to talk to users about how you can use these tools in your work. And overall, for my job, outreach is really about making our pathway data as accessible as possible. So we do provide our data to a lot of different resources. So you may be familiar with, for example, MSigDB or some of these other ones you see here. We provide all of our Reactome data to them so that they can also disseminate it to different users. So that's a part of my job, is to coordinate those strategic partnerships and provide different types of education, training, and direct user interaction. And yeah, that's everything I'm going to talk about today before I pass it off to Helia. So overall, we have a pretty large team right now of curators, developers, and PIs who help to manage this whole project. We also have a lot of contributors, reviewers, and collaborators, and a scientific advisory board. And of course, we are government funded, so we have to thank the NIH and other funding sources that allow us to continue working on this project. Yeah, that's everything I want to say. If you want to reach out, please feel free to email me or add me on LinkedIn and have a chat. Thank you.
All right, I'm going to share my screen. And you all see it, hopefully?
Yep.
Okay, perfect. So good afternoon, everyone. I'm Helia, and I'm a second-year master's student in Dr. Stein's lab. And I have been working with Nancy and everyone else in the team on creating a chatbot for the Reactome Knowledgebase. And I'm very excited to be here today and have the opportunity to share what we have so far. My project is called Navigating Molecular Biology with a Conversational Interface. Now, as we all know, as we dive deeper into molecular biology, the complexity of data we deal with increases exponentially, which means that researchers often spend quite a bit of time trying to extract meaningful information from the extensive breadth of information that is available across a multitude of biological databases. Now, our goal with this project is to streamline this process, making the information more intuitive and accessible through a conversational interface or a chatbot. So what you see over here is the current search interface of the Reactome database, where researchers typically type in keywords such as GATA6 to retrieve information that they're looking for. And again, as you see, the search results return various entries related to GATA6. However, it's important to note that these results often require quite a bit of manual filtering by the user to extract the relevant information that they're looking for, which can be quite challenging, and particularly for those who are not deeply familiar with the specifics of molecular biology or the data structure that Reactome uses. Now, comparing these results with I want to compare what we got from Reactome's current search interface with what we get from the chatbot that we've been working on. Now, as you see, with a simple query of tell me about GATA6, I got a lot of information along with links directing us directly to the Reactome pages as sources and citations, where users will be able to take a deeper look into the exact papers and the exact information that was used to generate those summations from Reactome. I am going to run another query just to show you guys in real life how the chatbot works, while I talk a little bit more about other features of our chatbot. We can again, for example, ask more specific questions, this time about NF-kappa B, and just as before, we would get a comprehensive overview of what NF-kappa B and the related proteins are, along with citations relating to it. Another very cool thing about it is that about this chatbot is that you would be able to ask questions in your mother tongue when your preferred language, and you would be getting results back in the same language, along with again citations leading you towards the Reactome database. Okay, moving on to create this chatbot, we have used RAG or retrieval augmented generation. And to deploy it, we have used LangChain quite a bit, but I will quickly walk you through how the system was set up as it is right now. So to create the system, we first retrieved and downloaded information about drugs, proteins, complexes, reactions, and pathways from the Reactome database. These information were then embedded and stored in a vector database, such that when a user asks a query or a question from the database about the database from the user interface, the question is sent to an LLM and is expanded, and this is to help capture a broader background information to allow the LLM down the line to give the user as much information as possible. The expanded queries are then used to search the vector database that we previously saved these information in, following the retrieval of the documents, since we are storing our files and our information for different databases and we're doing separate queries on them, following the retrieval step, we employ a re-ranking mechanism, also otherwise known as reciprocal rank fusion or RRF, which is integral to refining the selection of documents for response synthesis. And we're hoping that by applying RRF, we can enhance the relevance and the accuracy of information that is passed on to the LLM, which is critical for ensuring that the conversational interface can provide users with responses that are contextually rich and also grounded in truth. This information is then formatted into a prompt along with users' initial question, and is then passed on to the LLM to create to essentially generate a response. So far, to create this model, we've been using the OpenAI's LLM model and also the embedding models. But we have also tried using Llama, and we're still working on it. But the one that you just saw is using OpenAI. So yeah, that's what I have for you right now.
Cool.